Alright, so this is an additional video building on my last video discussing Griffith's experiment with mice. The controls of the experiment were the injection of the pathogenic S strain and the non-pathogenic R strain, which he did in the very beginning because they allowed him to see the effects of only the S strain and the R strains relative to the heat-killed S strain in the mix of the heat-killed S strain with the R strain later on in the experiment. The two strains were grown in separate cultures, and the R bacteria formed colonies that had well-defined edges and a rough appearance, which is why it was given the R abbreviation. The S bacteria formed colonies that were rounded and smooth, which is why it got the S abbreviation. And the smooth appearance is due to a polysaccharide coat, and that coat actually fended off the immune system, and that's what made the strain pathogenic. This was the first experiment that showed that bacteria can get DNA through the process of transformation, and this was shown by the DNA from the heat-killed S strain being taken up into the R strain bacteria. This is kind of like with the molecular cloning we discussed in class, where you input a gene of interest into a plasmid, and then that plasmid can be easily taken up by a bacterial cell.